Good morning, everybody. I'm going to present the methods that we have used in all the sub-challenges. We are a team, Team FAT, coming from 3 institutes, FIM, and Aalto University and University of Turku. From FIM, we are mostly bioinformaticians, and the guys at Aalto are working on the method that we've used for the sub-challenge 1, which is a Bayesian method. And then the guys at the University of Turku have developed the method that we've used for sub-challenge 2 and 3, which is called greedy RLS. Very briefly, I think we now already know about the dream challenge goals. So our goal is to predict the essentiality using the molecular features. And the sub-challenge 1 allows the most flexibility in terms of adding any extra features apart from the ones that are provided by the organizers. And challenges 2 and 3 require very precise number of features to be included in the final prediction models. And these descriptions have guided us in the model choices and the data choices. For sub-challenge 1, in addition to the mutation, copy number, variation, gene expression, we have used the tissue type, and then we have used 2 different kind of views, which are like pathway activity scores. So the MSigDB is, we have taken the gene sets from the MSigDB database associated with the GSEA, Broad Institutes, and using the gene expression data, we have computed the activity scores for each of the pathways. And then paradigm is another algorithm which computes the pathway activity scores using a, like representing the omics data as factor graphs. So like briefly about the pre-processing that we have done for different data sets here. In the copy number variation data, we have about 23,000 features. And we noticed that many of the rows have identical values, meaning that many genes have identical CNV profiles across all the cell lines. So in this case, we have simply used the first non-duplicate row among all the duplicate choices. So this is just done in order to reduce the dimensionality. And for the mutation data, wherever we have a missing mutation information for a gene in a cell line, we assume that it's not mutated. And then we have removed the rows which do not have any variance. And finally, we have normalized all the variables, input and output variables. For the sub-challenge 1, we have used the BEMKL method, and the motivation for using this method comes from the NCI DREAM drug sensitivity challenge, where many of our team members have participated and used this method. And so I won't go too much into the details of this one, but each of the data sources we use to calculate 1 or more kernels corresponding to each of the data sources. And there is an intermediate output variable called G, which is like a low dimensional representation for each of the kernels. And we have the kernel weights then, which are modeled to predict the outputs. So this method is like, it combines the nonlinear regression via the kernels and multi-view learning, where we can include many data sources, and multitask learning, which means that we are able to predict the multiple outputs simultaneously. Kirsten from Samuel Kaske's group has applied the BMKL for the sub-challenge 1. So this is like each, I'm just listing the views that we've used and the kernels that we computed. So once we have trained the BMKL, then we repeat the same process for the test sets. About the performances, one thing that we notice in sub-challenge 1 which is true for our group's performance as well as for the performance of all the other groups, is that there is a consistent improvement of the prediction performance over the different phases. So I think it's most probably coming from the increasing sample size. And actually this kind of trend is much more clear in sub-challenge 1 than in the other 2 sub-challenges. And in the final model that we have used, we see that like gene expression, copy number variation, mutations, and tissue type had like some kind of contribution to the prediction. Like both the submissions that were made corresponding to the first submission is an ensemble over the 5 folds of the cross-validation that we have used. And submission 2 is an ensemble over 3 different experiments where we have like tried several combinations. And one of the aspects that we have tried to explore is to incorporate the similarity of the gene essentialities that we have used in the following 3 experiments. And for the sub-challenge 2 and 3, since we need a very fine level control on the number of features that we choose, we have used the greedy RLS approach. RLS stands for regularized least squares. So here's a problem description of what this greedy RLS is. So Y is the output matrix, which is in our case the gene essentialities. We have M number of samples and T number of tasks to be predicted. T is like close 2,647 in both the sub-challenges. And molecular features are the gene expression, copy number variation, and mutations, where we have M samples, 105 cell lines, and D variables, where like we have approximately 45,000 variables. So the problem of greedy RLS is finding the matrix of weights, where we have D rows. So for each variable, we have a row of weights, and there are T columns for each of the tasks. And so the objective is to minimize the difference between X times W and Y. Subject to the RLS constraint, which is the L2 penalty, so like, and then the sparsity constraint basically means that we want to have at most K rows where we have 1 or more non-zero coefficients in the columns. So I define the regularization parameter here, which is basically just a restatement of the RLS and the ordinary least squares. So the equivalently we can define as finding W that minimizes the X W minus Y and the lambda times W squared. So this regularization parameter is a very key parameter. So then I come to the general algorithm of greedy RLS. So the RLS by itself does not give any sparse models, and that's why we need to explicitly consider how we come up with the sparse models here. The approach here is that we start with an empty set of features, and then we in each step, we add one feature at a time. And the way to add this feature at a time is to compute the leave one out performance for each of the tasks when we add that feature. And then we compute the average of the leave one outs over all the tasks. And repeat this process for each of the variables, each of the features, and then choose that feature that gives the best performance over all the tasks. So this one is repeated until we achieve the required desired number of features in our model. And finally, once we have found this set of features S, we just train the model. So this algorithm itself is like a very general one. Yeah, so it's like an approach for doing feature selection, a forward feature selection using the leave one out cross-validation approach. And the specialty of greedy RLS in this case is it uses like very specific computational shortcuts in matrix algebra to scale up the computational complexity, like scale up the computational feasibility. And so as a result of this, this can be done in linear time. In order to select the regularization parameter, which is a very crucial one, and in order to get a proper estimate of our performance, we use a nested cross-validation. Where in the outer cross-validation loop, so basically we split the data into 3 folds, and then in the inner cross-validation, we again split the 70 samples into again 7 folds. And so in the inner cross-validation, we try a set of 20 candidate regularization parameter values and choose the regularization parameter which minimizes the, which gives the best performance over the folds. And after that, in the outer cross-validation, we have then the performance estimation as an average of the 3 splits. So the sub-challenge 2, we have applied the same approach, where in the sub-challenge 2, we have t equal to 1. So the number of tasks is 1, meaning that we are predicting each gene's essentiality separately. So in our approach, we seem to have improved the performance over the phases, but actually the same thing does not actually hold for the best team. So there is an improvement of performance from phase 1 to phase 2, but there's a slightly little drop, even though it's not significant, from phase 2 to phase 3. So I don't know if this sample size can be directly translated here. And one thing I think is that we're not doing very good in sub-challenge 2, even though the method is actually designed to do the same kind of things. And I believe that we may need more than 10 features to be able to make a more accurate prediction. And so on the table, on the lower side of the slide, I want to convey 2 messages. One is that the performance evaluation that we get using the cross-validation is quite reasonable, in the sense that we get the same kind of performance also on the test cells. And the second thing that I want to mention here is that we try to also do a priori feature filtering before feeding the data to the greedy RLS, and it did not help us. So we have tried actually a couple of different ways of filtering the features prior to applying greedy RLS, and here I list one of the methods that we have used. So the method is so that we build a regular RLS with all the default parameter setting, which means we use the regularization parameter 1, which may not be the best thing to do, but the idea is to just get the ranking of the features. And once we get the ranking of the features based on the coefficient, we use the top K features, where here I have 1,000 features, 1,500 features, and 2,000 features, and so on. So in each of these cases, we see that the performance that we get is slightly lower than allowing the greedy RLS to choose the best features. Similarly, in sub-challenge 3, we have used again this greedy RLS approach. Actually, the way that I define the greedy RLS here is exactly designed for this sub-challenge. So here again, we have been able to improve the our prediction over the phases quite consistently, and also the best teams in the sub-challenge, in this sub-challenge, have been able to improve the performance from phase 2 to phase 3, but there's a little drop from phase 1 to phase 2, which is a little bit surprising. In terms of the features that we have selected, we have not retrieved any feature, any mutation feature into our model. So the feature selection did not get any of these, and we get 2 CNV features, out of 100 features. And this has been also discussed here and yesterday in the evening about not getting so many of these mutation and CNV features. And so like it was pointed out before, so I think that the gene expression is like an intermediate thing between the, so it's a kind of result of the mutations, copy number variation, and something else. So if we want to be able to utilize the data from mutations and CNV, probably we need to have some way to use the component of the gene expression which is not explained by the CNV and mutation somehow. And again, here also, the table shows that using the cross-validation, we have a very reasonable estimate of our performance, and the feature pre-filtering does not help so much. Actually, that thing does not come across so clearly here, because some of the feature filterings in our cross-validations seem to have done at the same level, slightly better, even though it's not significantly better. But in our test, in our final submission, we have the higher rank for the one where we did not do any prior feature filtering. And the second ranking submission we had was using the 5,000 features. So in a way it is good, but it's still worse than using all the features. And then we try to look a little bit deep into what are our predictions and so on. So it's actually nice to see Mehmet has already presented most of this stuff. So the thing is that there is average performance in this sub-challenge is like 0.23, which is, I think, relatively low. It's not really convincing, and but I think there are some genes which could be predicted fairly accurately. So as we can see in this histogram, there are some genes which have like 0.6, 0.7 correlation. And here on the right-hand side, in the bottom, I have listed the genes, the top genes which could be predicted well, where the correlation of the prediction was more than 0.6. And so I don't know any of those genes. So if there is some kind of biologist who would make, but of course, one thing here is that the prediction is these genes are the ones which could be predicted well using the molecular features. So these genes do not necessarily need to be the essential ones in the sense. So this is not, in that sense, maybe not so easy to interpret. And here on the lower side on the left, lower left side, I try to basically dig into the reasons why some of them could be predicted well and some so could not be predicted. And so this, on the x-axis, I have the variance, but there is not probably so high correlation. Again, finally, we try to also use the annotation of the gene ontology features. Again, we get some very general molecular functions, and we probably need to look more deep into these. So to summarize, the greedy RLS, which is a linear time algorithm, has given us the best prediction in sub-challenge 3. And we did not benefit from prior filtering of features. And more samples may potentially improve the predictions, as we have seen in sub-challenge 1 very clearly, and with our method in all the sub-challenges. And cross-validations have given us a very good estimate, and we have benefited from using the high-performance computing facilities of CSE, which was the institute that I mentioned in the beginning. And I think it was very exciting to work on the like an exciting biological question in the stream challenge and the unique data. So I thank the organizers for this. And